Hello friends, welcome to the second lecture of the series for studying the microbial cell culture and its application. So, in the last lecture we have seen four things or we have noticed that there are four steps in microbial culture techniques: nutrients of microbial culture, culture procedure, equipment for microbial culture and types of culture growth. And we studied in detail the nutrient of microbial culture. Basically, we studied also why biotechnology is important to begin with. Then for the growth of the microorganisms, basically what do we need? We need carbon, we need nitrogen, we need antifoaming agents, we need trace elements / cofactors, we need substrate. Then we also looked at what kind of substrates are there for laboratory scale where we do not need large quantities, and what kind of substrates do we need in for the industrial scale, such as and for what we have seen is that for the carbon source we need molasses, for the ammonium source we need urea, or nitrogen source we need urea, and then basically we have seen essentially the other requirements such as anti-foaming agents because of the production of foam and the loss of the product, and those kind of things. Now, today we will look at all the three things: culture procedures, equipment for microbial culture both at laboratory scale and the industrial scale, and the types of culture growth. So, all these three things we will look in today's lecture. In the culture procedure, so now you see we have a microorganism, okay, and we have before that we have a so microorganisms is needed for the inoculation. So, and before that we need a carbon source, nitrogen source, we have that, and now we want to produce our biomass or our product or metabolite. Okay. So, essentially we have from our equations that we have seen A going to B, we have A, and we have the starting point for B also for inoculation. So, we have everything that is required. Now, we need to know how actually to do it. Okay. So, that is where culture procedures come. In this case, two things are very important. Now, one is sterilization procedure and another is aeration and mixing. So, sterilization procedure is for ensuring that there are no contamination. Okay. So, because with microorganisms you do not see them, okay, you do not see them with naked eye, you cannot differentiate between microorganisms A or B. For example, you cannot differentiate E. coli and bacillus. So, you need to make sure that if you want to work with E. coli, only E. coli grows there, right, or if you want to grow bacillus, then only bacillus grows there and not other microorganisms come into the picture. Okay. And as you can imagine that sterilization procedure is much simpler and easier when you are working with smaller volume, let us say 100 milliliter or 1 liter. It becomes complicated when you start to work with 100 liters or 1000 liters. It starts to become even more complicated when you have holes in between and there is an intervention, like for example, you are supplying air or you are supplying some new media on a continuous basis, right. So, these are the things that we will discuss in the sterilization procedure. In the aeration and mixing, and well, in our case or in the case of fermentation or product formation, we need aeration. Continuous supply of air is actually required, and why it is needed? It is needed because microorganisms use that air. So, they also breathe this air, not in terms of breathing as we do, but they also use air as an oxidation process and use this air to degrade the substrate and convert the substrate to biomass or any other product of their choice. Well, that is the requirement of oxygen or air for the microorganism, but why aeration is required is that in the reactors the presence of air is not sufficient. The head pressure or the air that is present just at the top of the liquid is not sufficient to cater for their all needs. So, we need to basically do an active flow of air. So, that is called as aeration through pumps. Okay. But again, in a smaller reactor or 100 milliliter flask, there is the air that is at the top of the flask. If you have a 250 ml flask and you are using 100 milliliters working volume, the rest of the volume is occupied by the air, and that is enough for you to carry out your process and enough mixing and the top and mixing is enough, and you can mix them, and it is actually it works well, it is fine. Again the same thing for the mixing. In the smaller flask, mixing is easier. You can manage the mixing quite easy, and every nutrient is available, is homogeneously distributed, right. While in the larger reactor it might be complicated, and that is why we need specialized equipment for that, and this also we will see in the today's class, all right. So, for sterilization, as I said, the sterilization procedure is for the removal of microbial contaminants. Okay. Why we need to remove the contaminants? As I said, you need to grow the organism or your process with E. coli, and you do not want any other microorganisms, be it bacillus or any other microorganisms to be present there. Similarly, if you want to grow with bacillus, you do not want to grow E. coli or the microorganisms, okay? And it has a significant effect. For example, I will give you a very small and interesting example, and this is not directly related to the bioproduct formation, but anaerobic digester systems. So, in anaerobic digester system, basically you have no oxygen present, okay? And what happens is that your substrate, what you add, it goes to form methane, okay, CH4, and these microorganisms are methanogens. Okay. I will just write a short form. Okay. Now, for instance, in this case, if you have a small amount of sulphate present, that is SO4, and you have SRBs. What are SRBs? SRBs is a group of microorganisms which are called as sulphate reducing microorganism. It is a group of microorganisms. There are many kind of microorganisms in this. Okay. Similarly, methanogen is not one microorganism, it is a group of microorganism. If sulphate, it produces sulphide. So, basically S2 minus. Okay. What happens is that if, for example, you have SRBs present in the reactor, what will happen is that this SRBs will take up this substrate. Okay. So, what this will lead to is that no production of methane. Okay. Similarly, for example, if you are producing ethanol, okay, and the microorganisms or yeast that is used commonly is Saccharomyces cerevisiae. So, you have a substrate which can be, for example, glucose simple, and it goes to ethanol. Okay. The microorganism that is there is SC, Saccharomyces cerevisiae, it is a yeast, or PP, Pichia pastoris. Okay. In case of this two microorganisms, there is some other yeast, okay, then your other yeast or some other contamination, then your ethanol will not be produced. Okay. So, and you cannot really, another challenge in this is this, okay, that you cannot really check whether easily whether it is E. coli or bacillus, okay, whether it is E. coli, bacillus or any other microorganisms quickly. You need to go to genomics, you need to study their DNA sequences to really find it out. Okay. So, what, and if you are running a reactor and even if it takes 6 hours, a 6 hours of downtime in a bioreactor causes money, you lose money. So, that is why what you need to make sure is that your sterilization procedure is accurate, and that is why it is very essential that we should remove the microbial contaminants. All right. Now, how we do it? For sterilization procedure, how to do it, basically there are, at a different scale there are different ways. Okay. In the laboratories, the most easiest way to do it is with autoclaves. Okay. So, what are autoclaves? Autoclaves are machine that heat up with the steam thrown in the reactor, and in the machine itself we basically add the steam in the reactor to heat the reactors to a temperatures higher than 100 degrees at a pressure of 15 pounds per square inch, that is 15 psi. And basically this is a recommended value. This is a value that actually there are reasons to select this 15 psi pressure or pounds per square inch pressure and the higher temperature. Okay. And the reasons are as follows. Because it has been seen that spores survives anything less than that. Okay. So, spores survive anything less than that. And what is the temperature here? So, temperature is normally 121 degrees. Okay. So, why not at 100 degrees and normal atmospheric pressure? No, what we do is basically we increase the pressure so that the boiling point of the water increases. So, I think you know this thing, as temperature increases, okay, sorry, this should be as pressure increases, okay, this will lead to T boiling point increases. Okay. So, normally at 1 atmospheric pressure, okay, 1 atm, the boiling point is 100 degrees Celsius of water, right. When we have 15 psi, you can convert it, it is not that difficult, you can do it, the conversion between psi to atm, atmospheric pressure, it is easily done. At 15 psi, basically the boiling point increases to 121, 122 degrees centigrade of water. Okay. So, what happens? What happens is here is essentially that we are giving more heating power or more thermal energy to the water. Another important thing to note here is, I think this you would definitely know, whether a water at 100 degrees or steam at 100 degrees, which has higher thermal energy. This one. Why? I think you know something about latent heat of vaporization. So, a water at 100 degree eat up this latent heat and then convert to steam. Okay. So, that is why we basically have this latent heat of vaporization, and that is why we use this steam, and at this, with our experience we know that that essentially at 121 degrees 15 psi pressure for 20 minutes we can first reach, it is enough time for the steam to reach everywhere, and then again enough time for the steam to basically kill everything. Okay. And then your media or whatever you have is completely autoclaved. Okay. Is completely is autoclaved, and then that means basically it is completely sterilized. Okay. So, this is how basically we do it for 100 or 1 ml or 1 liter flask, even 250 ml flask, and so on and so forth. But what about large reactors? Okay. How do we deal with the large reactors? Well, for the large reactors what we do is basically we put steam. Okay. So, we basically have reactors that have jackets on the top of them. So, jackets means a curl of our tubes that can pass, that can heat up the reactor, and in that reactor we put in that place we put basically steam to increase the temperature of the reactor to 121 degrees, with 15 pounds psi for again 15 to 20 minutes. Okay. So, basically that is how we will autoclave the large reactors, and including tubings. Okay. Everything that will come in contact with the media A, so the nutrient growth media or the biomass. Okay. So, for example, if you want to give air, you also need to sterilize the air before you feed in. Okay. And of course, you cannot heat the air, right? Means it is not possible that you heat the air to autoclave it. So, for air you basically use filters, filters that are basically connected to the tubes, and these filters are sterilized. So, they have never been used, and basically they will allow only air to pass through, and no viruses, no microorganisms, nothing, because the pore size is very small. Okay. So, what is filter sterilization? Filter sterilization is something like this. Okay. Let us say this is our filter and this is our pores. Okay. And you have microorganism that wants to go out in, and then you also have the crosses is air that wants to go in. Okay. What will happen is you have cross that goes in while microorganisms would stuck here. So, anything in this region it is sterilized. So, we use this for air, yes. What else? We also use this if you want to add vitamins, okay, or if you want to add some enzymes. Okay. These enzymes are not typically added for fermentation process, but in other processes in case you want to add it. Why? Because these are heat sensitive. What would happen is that if you heat them at 121 degrees Celsius with at 15 pounds psi 15 psi pressure, okay, then essentially what would happen is that they will not be able to they will get deteriorated or degraded, and once that happens they will basically not be stable anymore. Okay. So, that is about the uh, culture process and sterilization. The next is aeration and mixing. Okay. So, as I said, why we need mixing? Mixing is needed because you want to mass transfer of oxygen and uniform presence of media components. Okay. And as I said, it is easier in a it's easier in the flask, okay, smaller volumes. It becomes increasingly challenging in the large reactors, and for that you need specialized equipment, and as I said, this will again study in this lecture only. Okay. So, what when I say what mass transfer, what do I mean? I basically mean something like this, that you have a small, let us say not a small, but whatever sized flask or a reactor, okay, and this is what you will say homogeneous mixing. Okay. Homogeneous mixing or rather homogeneously mixed. Okay. While in other case, if you have something like this, okay, this is not homogeneously mixed, right. Okay. And you do not want this, you want this. Okay. So, essentially we want homogeneously mixed situation and not homogeneous mix or heterogeneous mixing. Okay. And for that this is required because if your microorganism is growing here, let us say at this point, okay, if your microorganism is growing at this point or this point and this point, okay, then it is fine, it can grow anywhere. But if your microorganism is growing this one and this one, basically this would be at disadvantage as compared to this, because it has more media and component while it does not have. Okay. So, that is why we require homogeneous mixing, and once you have homogeneous mixing, you also have higher mass transfer, because your mass transfer. So, what do I mean when I say mass transfer? When I say mass transfer, that means component A, not from our reaction, let us not use that one, X is going to a, a, a spatial distribution, or essentially X is going to place A and X is going to a place B. So, this is a place, okay. It is a in a spatial distribution, this is a place A and this is a place B. Okay. So, when I say mass transfer, that means basically the X is moving to A and X is moving to B, and this should be the same, it should not be the different. Okay. And this X moving depends on concentration of X. Okay. So, when it depends on the concentration of X, that means if I have higher substrate concentration at the bottom, that means the mass transfer would also be higher, while when I have a smaller concentration at the top, the mass transfer would also be lower. Okay. So, that is what is required for effective mass transfer, and thus we need good mixing. Okay. One important thing is that so we will see how we do the mixing in the next part of the lecture today. One important thing is that aeration, like you are providing air, that also helps in mixing. How it does is this: you have something like this, a reactor, okay, and you have supply of air, and air is coming like this. Okay. So, when air, because it is lighter than, this is water, lighter than water, it will move up, and when it moves up, it also takes with it biomass. So, because biomass has a tendency to settle down, while air has a tendency to go up. So, let me draw this in a bigger figure. Okay. This is your water, okay, and this is your sparger coming to supply air, okay, and this basically is air. So, air would go up. Why? Because air is lighter than water, okay. This is lighter, while the biomass which is here has a tendency to come down, because it is heavier than water. Okay. So, since it is heavier than water, it will come down, while the air which is lighter than water will go up. So, this will come up down, and this thing would go up. So, they interact, and then because of that the aeration also causes mixing. Okay. So, now let us see basically what kind of equipment you require. So, you know that you need sterilization procedure, you need mixing, okay, you need the equipment to withstand sterilization procedure. So, that means they should be able to handle 121 degrees, 15-20 minutes and around 15 psi pressure, okay, and then they could survive this thing, right. So, we also need to see what kind of equipments normally we use. Okay. So, one of the, these are the three equipments which are used. Another one is autoclave, but autoclave is not directly related for microbial culture, it needs for processing of the media. Okay. For mixing, we at the lab scale we have baffled flask, and this baffled flask when put on shakers and incubator shaker, you can get the mixing. and for other things we have fermenters. Okay. You can have fermenters of different sizes, laboratory scale, pilot scale, large scale, industrial scale, and so on and so forth. Okay. So, we will have a look at it. So, these are the baffled flask. You can have normal flask as well, and you can also have baffled flask. In the baffled flask, what you see in the arrow is there is an indentation. So, there is basically a curved in structure, and what this does is that it causes turbulence of the agitated culture medium. Okay. So, if the culture medium is, if it is smooth, then the culture medium would go in a normal direction, that there is no basically disturbances, but as you cause the disturbances or the turbulences, this leads to better mixing. Okay. So, it is something like this, that you have a water that is flowing, okay, and you throw a rock in it suddenly in the direction and the opposite direction of the water, or a structure that comes, like for example, how you go about in river rafting. Basically, the water is getting turbulent because of the many rocks and the structures that are coming in place in between them, and it is affecting the flow, and that basically is creating your turbulences. Okay. So, this is something that we also do to ensure better mixing. Okay. How does the oxygen come in the baffled flask? Well, if you have a 100 milliliter, or let us say a 250 milliliter flask, you do not fill it completely with media. You fill it only till 100 ml with media, okay, and the rest of the thing is you will keep it open, and that is actually filled with rest of the thing is filled with air, and this air is also can be used for the aeration requirement of the culture. Okay. So, what is the typical procedure? The typical procedure for running experiments in the baffled flask would be something like this. You have a flask, okay, here is an indentation, okay, and this is 250 milliliter flask. What you do is you fill it with your media, okay, till here, okay. You close this over here, okay. So, you close it with either aluminum foil or cotton plugs, okay. Then you pack it and you autoclave it. So, essentially sterilize it. Once you have sterilized it, then you take this, okay. This is your liquid volume, okay. Rest is air, okay. This air is also autoclaved now, right, because it has gone into the autoclave along with the media, and there is no mass transfer or air transfer in this zone. Okay. So, no air from outside coming inside and no air from inside going outside. Okay. So, that is how you autoclave, and once you have autoclaved this, okay, you can go into another thing which we are not going to discuss, but called as a box. It is called as laminar chamber. So, and there basically you can open it and it is completely sterilized. You can add things in it without getting it contaminated, and you can also work near flames, like really a flame like this, like a fire, okay, or like a candle, but bigger, not that small, a little bit bigger, in a laminar hood, and then basically you can transfer your inoculum and so on and so forth. Okay. So, that is how an experiment with this would look like. I have talked about closing the shake flask with aluminum foil, but you can also many times use cotton plugs actually, and cotton plugs actually can provide more air. So, aluminum foil is completely blocking the transfer of air from outside to inside, but cotton plug actually can provide the air from outside to inside, but at the same time sterilize them using a filter. So, they can act as a filter, and they will not allow any of the microbes that are present in the air to go through and contaminate. Okay. So, that is why sometimes you prepare the media, you pack it with cotton plugs, and then you autoclave it. Okay. So, So, that is also one of the other method to pack the media is. So, now we have autoclaved and sterilized our media, we also need to put it on a shaker, so that it can have a mixing. Okay. So, and we need to do a mixing. So, to do that basically we put it on a shaker, and this is a shaker that you see on the screen A. So, this is a shaker, what it does is it will have a rotation, it will rotate, and basically let, since the liquid is there, liquid would also move and it will ensure mixing, and it works quite well. This is the first experiments that if you these are the first experiments that you do in the biotechnology. Okay. Growing something in the culture medium and on shakers. Okay. What you can control is the RPM. Okay. And it is normally the temperature is at room temperature. Okay. But for example, in some cases, for example, E. coli grows very well at 37 degree. So, what you do is you have an incubator shakers, which is B that you see on your screen, and B image that you see. Okay. And this incubator shaker is something, basically it is a closed chamber, again it will rotate and things like this. Okay. But it can also control the temperature. Okay. So, you can increase the temperature to 37 degrees, 30 degrees, 35 degrees, whatever you like, and rather whatever your microorganism like. Okay. So, this is how we ensure mixing in the laboratory scale. What about full scale? Well, in full scale, industrial scale, you use fermenters, but that is not to say that fermenters or bioreactors are not used in the laboratory scale as well. To basically run a 1000-liter fermenter, you need to run maybe 0.5 liter, 1 liter, 0.5 liter, 1 liter, and then 100 liter in between. You cannot directly take shake flask experiments and run it in a fermenter, because it is a completely different setup, and microorganisms would not like that kind of behavior. Okay. So, these are the equipments for fermenters or bioreactors. So, this is how a bioreactor and fermenter looks like, and this you also studied in your last class, not in this one, but in the previous units. So, what we have essentially, and I will just give a quick overview. What we have is, the important thing to look at is the impeller. Okay. So, impellers are the one that rotates, and because of this rotation they create turbulences, and because of this turbulences they create mixing. Okay. So, that is one thing. Then another thing to look at it is, you have an cooling jacket, okay, as well as a steam supply on the top. So, cooling jacket is to decrease or increase the temperature or cool the liquid, while steam is for autoclaving. Okay. Then what you have is essentially at the bottom you can see spargers and air bubbles. So, sparger is something that actually dissipates the air, and this sparger is fed with a sterile air. How do we sterile air? I told you, we just put a filter there and keep changing that filter from time to time or batch to batch. Okay. So, the air is coming, that is clean, then impellers we have already talked about, and you have pH controllers and acid and pump reset and pumps to basically add acid and base to it. Okay. So, these are the things that are there typically in a fermenter or bioreactor. Okay. So, why do we use fermenter or bioreactor? Okay. This, we use them for the large scale growth of microorganisms under a controlled environment. Okay. If you can, as you can see in this fermenter or bioreactor, we can control aeration, that depends on the flow rate of sterile air. We can control agitation, that depends on the RPM of our impeller. Okay. We can control temperature, that basically depends on the cooling jacket, okay, or the steam if you want to increase the temperature, and then basically we can control the pH. As you can see, there is a pH probe, and it is a dynamic control of pH, that if there is pH is going in some other direction, acid would be pumped in the acid or base would be pumped to bring the pH to the desired value. Okay. We have a drain and overflow, that essentially you can So, once you are done with the process, you can just open the impeller and take your harvest, or if you want, you can also operate it on a continuous basis, and this will see how the things are operated in a batch mode, fed batch mode, or a continuous mode. Okay. Of course, when you run such a thing, okay, so if you are running 100 milliliter or 250 milliliter in a shake flask, you are still is okay. If something goes wrong, it's fine. It's not such a big loss. But for example, if something goes wrong, a big reactor like a 1000-liter reactor, you lose two things: time, because it takes time to start a reactor, and it causes your production, the total amount that you are going to produce in a particular period of time will come down. So, it causes loss in money, right. And the second thing is that basically it causes you loss in the material as well, because losing 200 milliliter is okay on one hand, losing 1000 liter is another ball game. So, basically for that what we have is sampling ports. So, you can sample from time to time how is the reaction going on, and if there is some intervention that is required. Okay. So, that is why it has equipped with sampling port. Okay. And it is also required to do the sampling port also to see if there is a problem in mixing, because then you can take samples from different height of the reactor and see if the biomass concentration or your metabolite concentration is the same or different, and things like this, to know if the mixing is fine or not. Okay. So, this is how a bioreactor would be set up, and what you see in the figure is a most commonly used stirred tank reactor. Okay. There are other kinds of reactors as well. There are bubble column reactor, fluidized bed reactor, photobioreactor, and they have different principles and different advantages and disadvantages. Okay. It depends on your process, the kind of reactor that you will use. Okay. But we will not go into the detail of those. For the sake of this lecture, I think stirred tank reactor and understanding that a reactor would look like this is, I think, is enough. Okay. So, then we move to some figures to show you how the reactors look in general. So, on figure A you can see a laboratory scale fermenter or bioreactor. Okay. So, essentially it's a tube, and it's basically a tank, a cylinder, and then lot of tubes going in and out of it with motors and impellers and things like this. Okay. On the other hand, if you see figure B, you see industrial scale fermenter. Okay. Just see the size difference. Yeah. So, you see that basically the industrial scale fermenters are really big, and this is a normal procedure, because the quantity that you produce is much higher in the industries, and it requires to be higher, because if you produce at the laboratory scale, the economics will not match up, and the cost would be very, very high. Okay. So, the industrial scale fermenters are 100 liters to 4000 liter. 4000 liters is a really big one. Typically it is 300 liters, 1000 liters, and so on. Okay. In the laboratory scale, 10 liters to 100 liters is okay. Even smaller are better, maybe 5 liters, 50 liters. These are the ones that are normally used. And generally, to really do the research part, you use smaller reactors, 1 liters, 2 liters with a working volume of 1 liter, and these are relatively okay, because you need to, when you want to study process in a continuous reactor or in a reactor as compared to baffled flask, you really need to operate it in the reactor, but at the same time you do not want to operate for analysis of 5 milliliter, you do not want to run a 100 liter system, because you would need to run that 100 liter system or that system many a times to really find out all the characteristics of that system. So, essentially what you do is you run a smaller volumes. So, 0.5 liter, 1 liter, this is a good volume to do a study. In some cases, in laboratory scale, if they want to use a product for research purposes, such as that I want to produce a compound or a metabolite and I want to use it for my research purposes, then you do not run 1 liter, you maybe run 10 liters or 50 liter reactor, so that you produce it enough quantity your metabolite for your research purposes. But again, that is not an industrial scale. For production at industrial scale, you really go above 100 liters or 1000 liters and so on. Okay? So, that is the equipment for microbial culture. Now, let us look at types of culture growth. There are 3 types in which you basically run the cultures, is batch culture, fat batch culture and continuous culture. Okay. What is a batch culture? Batch culture is that you add everything, you seal it and you forget it. After 2 days, 3 days, 5 days, 7 days, you come out, you take your metabolite, you analyze everything, that is batch culture. What is fat batch culture? Fat batch culture is you put everything and after every 8 hours, 10 hours, 12 hours, whatever time or 1 day, you add fresh nutrient and then you seal it, then you run again for a day, then you add another fresh medium, then you seal it, and like this you run it for 7 days or 5 days till the volume has increased that your flask no longer can take that volume. What is continuous culture? Continuous culture is, as the name suggests, you run it continuously. It can run for 7 days, 10 days, 20 days, 100 days, no problem. You can keep on producing as long as you want. Of course, at certain point of the time, theoretically it can go unlimited, but at certain point of the time your machine would need maintenance, there would be some problem in the culture and so on and so forth. So, that happens. Okay. So, let us look at the batch culture. So, these are the characteristics of the batch culture. Okay. X is your cell density, S is your concentration of substrate and QS is cell specific substrate turnover. So, QS is actually dependent only on S, it follows similar profiles like S. What are examples of this? Your shake flask is a typical example of this, but you can also run batch reactors and it basically demonstrate the complete cell growth phases. So, you will have if you run it long enough, you will have lag phase, then you would have log phase, then you have stationary phase and then you would have that phases. Okay. So, what does a batch culture means? Okay. Why? So, I will just draw for your convenience here. You have time, okay. Your x goes like this, your QS comes like this and your substrate would also come like this. Okay. And this is not that difficult to understand. Okay. I will show you how. Let us go to our characteristic equation, A goes to B, simple. Okay. What happens here is A in the case that with time. So, let us say this is at 0 hours, this is A1, A0, B0 at time 12 hours. Okay. Your A is let us say A12 and B12. So, what can we say about this? We know that A is getting converted to B, that means A12 would be less than A0 while B12 would be greater than B0, right. Similarly, if for instance, I do it like this, T goes to 24 hours, I will have A is 24 and B is 24. That means A24 would be less than B less than A12 less than A0. Similarly, B24 greater than B12 greater than B0, right. That means A is our X, A is our S would come down with time, B is our X would go up with time, right. So, this is what batch culture is. Okay. And essentially it makes sense that A goes to B and then A goes on depleting and B goes on increasing. That means, S goes to X, S keeps on decreasing while X keeps on increasing. Okay. So, I think that is pretty clear there. The only thing here is that it is not such that X would go on to increase infinitely. Okay. Basically, X would, so X would typically follow like this. Okay. This is lag phase, this is log phase, this is stationary phase, and this is that phase. Okay. And basically your X S would be somewhere something like this, right, completely ending. So, this would be somewhere S. So, X cannot increase infinitely because S at certain point of the time would finish, and that is the end of the fermentation time and the bioreactor time. Okay. What happens in the fed batch? So, in the fed batch, this is how the graphs looks like. So, here you see the concentration of S is constant. So, as Qs is dependent on S, that is cell specific substrate turnover rate, Qs also remains constant with time, but X increases, right. What is X? As we have seen in the batch, the denominations are the same. X is your cell concentration, S is your substrate concentration, and Qs is your cell specific substrate turnover rate, right. And this also makes sense, right, because what is happening is, so this is the volume at time is equal to 0, this is the volume at time is equal to 12. Okay. Basically, this is the volume at time is equal to 12. Okay. So, this one, okay, same volume, and this is the volume at time is equal to 12.5 hours. So, instead of this, this has increased till here. Okay. So, here basically you had A0, B0, here you had A12, B12, here you had A12.5, B12.5. So, I can say like this, right, and this is our characteristic equation, A goes to B. I can say like this, A0 is more than A12, okay, right, but can I say A12 is greater than A12.5? I cannot say this. Why? Because I have added fresh A here, that is why my volume has increased. Okay. So, now imagine if I am adding this A, okay, very slowly, that would mean that whatever A is consumed, I am replacing it. Okay. That means my A concentration, or in this case S. So, whatever S is consumed, I am replacing it. That means whatever S I am using, I am replacing it, and hence my S concentration remains the same. And that is why also Qs, which is cell specific substrate turnover rate, also remains the same, but my X, which is growing continuously, would remain. When would this fed batch culture would stop? When you have achieved that the volume of your starting reactor has been there, and basically it cannot be increased any further. So, at that point of the time, you have to end up the fed batch culture. Okay. The next is continuous. Okay. So, characteristic of the continuous is actually quite easy, no changes in x. So, this is your continuous culture. The characteristics feature of the continuous culture is the same, no changes in x, no changes in qs and no changes in s. Okay. And what it means is that basically one nutrient is, let us say, is limited and it is added continuously as it is about to finish. Okay. And because you are adding the volume, you whatever volume you add, you also take that volume out. Okay. So, that means the volume of the reactor remains constant. Okay. So, when you are taking that volume out, basically you are also taking your substrate S is consumed to form X, and whatever extra X is formed, it is being drained out, okay, in the extra volume. But when it is draining out, the because of the fresh substrate, the new X is formed. Okay. And this is what a continuous culture would mean like. So, if you look at this, this is your reactor, you have S going in, okay, and in your reactor you have concentration of, this is your S0 going in, this is your S, let us say S1 and X1. The thing going out would be S1 and X1, right, because it is completely mixed system. Okay. So, essentially what is happening is that whenever S0 or whenever this S1 starts to deplete, okay, fresh S0 is added, okay, and this is added on a continuous basis. So, it is going on a continuous basis, okay, and as soon as basically your biomass leaves, okay, so you have a flow rate of, let us say, 10 liters per hour, okay, but which is quite high, you might need a smaller flow rate, maybe 1 liter per hour. Okay. If you have flow rate of 1 liter per hour and that is the volume of your reactor, then basically at 1 liter per hour X1 concentration biomass is going out and the same amount of biomass is produced in that 1 hour in the reactor. So, that your concentration of X does not changes, concentration of S does not changes because S is replaced by the fresh media, while X is lost due to the biomass going out in the reactor is replaced by fresh production of X. Okay. So, where is we are losing S? S is being lost because it produces X, and why we are losing X? X is because its output, it is going out. Okay. And how this S is replaced? Fresh media. Okay. So, input. And how is this X replaced in the reactor? Basically X is produced. So, this is your continuous culture. Okay. There are two types of continuous culture. You have a chemostat where the chemical environment in this reactor is constant. Okay. Basically that means essentially all the chemicals that are there, it remains constant. So, the chemical composition of the cell also remains constant, okay, RNA, DNA, this kind of things. In turbidostat, basically your cell concentration remains constant. Okay. Important thing to know is that when you start that reactor, okay, it is not as if that as soon as you start the reactor everything is working well. The reactor will take some time to essentially come at a point where no changes in X, S or Qs will happen. Typically, it will look like this. So, this is your reactor, this is coming in, coming out, okay. And if you operate this reactor with time, it will go like this and then it will continue like this. Okay. This can be your x y axis can be your X cell concentration. If it is S, then your S would be this. like this with time. From here point onwards, you can say that the reactor is at steady state. Okay. And when the reactor is at steady state, it means all the concentration in the bioreactor remains constant. Okay. With that, we will end this lecture. Thank you very much, and we will continue in the next lectures the microbial cell growth, how do we measure it, and look at the kinetics. Okay.